Welcome to EPG Pathshala and the second module on sequential clinical trials. In the first module, we have generally looked at the general framework for such designs and seen that it involves some key components. The first being the parameter, the statistic, based on some underlying expectation of our behavior of the statistic in probabilistic terms. The stopping rule, post-trial analysis, implementations in terms of continuous plans, adjustments such as the Christmas tree adjustment, and finally, post-trial analysis. In this module, we shall concentrate more on the concept of interim analysis and see why it is necessary as well as what statistical implications it has. Now, the idea behind interim analysis, of course, is that we have a situation where patients are being recruited over time, and hence patient recruitment could occur gradually over a very long period of time, even as the trial continues. The standard procedure, of course, is to look at the results at the termination of the trial, but there are ethical considerations which say that if we have conclusive evidence, it is necessary to stop the trial and to ensure that we use a procedure which guarantees that more patients are allocated to the better design. However, note that there is a problem with establishing that the evidence is conclusive, and in particular, the effect of performing interim analysis is to increase the overall level of significance. This is because the probability of finding a significant difference is now no longer that we find it at the very end, but the probability that we find it at least at each interim analysis. Thus, if we conduct a test of significance with significance level alpha at each interim analysis, the overall significance analysis, which is the probability of the corresponding union, is guaranteed to be larger than alpha. In this module, we shall look at methods for adjustment for alpha. We shall define something called the nominal significance level, and we shall see that there are different procedures depending upon whether we want a constant nominal level or we wish the nominal level to vary over time. Learning objectives for this module. We shall first define the concept of an interim analysis and also look at its importance and the effect it has on the statistical properties of a trial. We shall distinguish between the nominal and the overall levels of significance. We shall compare group sequential designs to sequential designs which assume that it is possible to perform an analysis after every single observation, and we shall also see that it is possible to define these kind of rules for alternative responses, that is, alternatives to the usual assumption of a continuous outcome. Second module on sequential clinical trials. This module will generally be based on two papers. The first is entitled Interim Analysis for Randomized Clinical Trials, and it is due to Pocock and appeared in Biometrics in 1982. The second paper, which is also due to Pocock, is entitled Group Sequential Methods in the Design and Analysis of Clinical Trials, and it appeared in Biometrica in 1977. We shall also refer to the paper by Armitage in 1969, which derives results for continuous sequential plans, that is, sequential plans where it is possible to analyze the data after every single observation or every single pair of observations accrue. And although this might not be logistically feasible in practice in many situations, the calculations from this situation form the basis for which we are able to derive the properties and notably satisfy the power requirements in the case where we have group sequential designs. In interim analysis, our purpose is thus to determine whether to stop the trial early because of a substantial treatment difference. So our situation is that we have patients who are accumulating over time, and the accumulation occurs gradually over a long period of time, and hence, even as the trial continues, we also continue to have a body of evidence. Our position is that it is possible that we have conclusive evidence even before we have concluded the trial, and hence it would be ethical to stop the trial because we would wish to allocate more subjects to the better treatment. Group sequential designs typically have the following three components. Firstly, they specify the maximum number of analyses. So here we do not analyze the data after every single patient or every single pair of patient accrues, but we assume that we should look at the data a maximum number of times. We shall apply repeated significance tests to the cumulative data. That is, at each interim analysis, we shall apply a test of significance to the cumulative evidence up to that point. And stringent nominal significance will be used as a stopping rule. Now note that the effect of performing an interim analysis is to increase the overall level of significance. This is because the probability of finding a treatment difference now is the union of the events that we find a significant treatment difference in at least one of the analyses, and thus if this probability is assumed to be alpha for each analysis, then the overall probability must necessarily exceed alpha because it corresponds to the probability of the corresponding union of events. The way around this is to define something called a nominal significance level, which is much more stringent than the overall alpha which we desire, and to use this as a basis for a stopping rule. Power calculations can be performed, and these are based on numerical integration. It has been shown by Pocock that there is little advantage beyond 5 interim analyses. This is based on considerations of the average sampling number. And finally, there are also alternatives to the fixed nominal significance level, in which we can have the nominal significance vary as a function of time. That is, we could have more stringent significance levels at early stages and less stringent levels at the last 1 or 2 analyses. So once more, our background is that there is entry of patients into a randomized clinical trial. The entry is gradual. We wish to perform interim analysis while the trial is in progress, in case the evidence is sufficiently convincing even before conclusion of the trial. Our assumption here for a sequential design would be that the data can be studied continuously and the stopping rule applied immediately at any point of time. This is the assumption which is made for a sequential clinical trial. However, this assumption may not be feasible in practice, and hence, as an alternative, we consider group sequential designs. Armitage, in his 1975 paper, describes various forms of sequential designs and analysis of clinical trials and tests of repeated significance. This paper is largely based on the papers by Pocock in 1977, and the results are obtained by numerical integration. For more details, you are referred to the paper by Armitage, McPherson and Rowe in 1969 and McPherson in 1974. Many of these procedures use two-sided significance tests, but you can look at the paper by Demetz and Ware for the case of one-sided testing. Now note again that the effect of repeated significance test is to increase the overall significance level or the type 1 error. So for example, Pocock shows that 10 repeated tests will increase the overall significance level from 0.05 to 0.19, and with more repeated testing, one can be increasingly sure of declaring a significant treatment difference whether or not one is present. For more details on this, you are referred to the paper by Armitage. This has been done for the normal setting. The way around this is to choose a more stringent nominal significance level for each repeated test performed at each interim analysis. Two key decisions have to be made. The first is how many significance tests should there be, that is, what is the maximum number of groups or the maximum number of interim analysis that we are willing to perform, and how many patients should be evaluated between any two tests, that is, what should be the size of each group. So basically, in a nutshell, two decisions: what is the maximum number of groups and what is the size of each group. In this table, we look at group sequential designs for a normal response assuming a known variance sigma square, overall significance equal to 0.05, power 1 minus beta under HA equal to 0.95, and several choices of capital N, which is the maximum number of groups. The results here have been obtained by numerical integration. So note that in general, the required number of patients per group will not be an even integer, but in practice, this rounding problem will have a negligible effect. For any choice of alpha, beta, and delta by sigma, the standardized difference in mean, as the number of groups capital N increases, the number per group 2n decreases, and the maximum number of patients 2nn increases. A very important piece of information of a group sequential clinical trial is indicated by the last column of this table, which is the average sample number or the average number of patients until the trial is stopped under HA. So note that there is very little reduction in the ASN in going beyond a 5-group design. So based on this, it would seem that there is little gain in choosing a design with more than 5 interim analyses. On this slide, we look at the comparison with continuous sequential designs. In general, the statistical literature on stopping rules has been predominantly concerned with continuous sequential designs. In this table, we see the maximum number of patients and the average number of patients under the alternative for the fixed design and various sequential and group sequential alternatives. Note that all the sequential and group sequential designs will increase the maximum size of the trial but reduce the expected size if the specified alternative hypothesis is true. It is well known that the SPRT open sequential plan has minimum ASN under HA, but since it has no finite maximum number of patients, it is generally considered inappropriate for clinical studies. On this slide, we are looking at the group sequential design for capital N equal to 5, alpha equal to 0.05, beta equal to 0.95 with varying nominal significance levels which are chosen to minimize the ASN under HA. For type 2 error beta equal to 0.5, the optimal design has nominal significance levels which increase markedly over the 5 tests with a consequent reduction in ASN under HA. Note that the optimum design for 1 minus beta equal to 0.9 or 0.95 does not have monotonically increasing alpha dashes, that is the nominal significance level, Hence, for 5 group designs with reasonably large power, 1 minus beta equal to 0.9 or 0.95, the choice of constant nominal significance levels is near optimal as regards the ASN under HA. Calculation results here are largely based on those used by Armitage in his 1969 paper. So here we assume that we have a series of observation x1, x2, xn and the random sum Sn equal to summation I1 to n xi, a sequence of cumulative sums. We stop sampling when for the first time mod Sn is greater than equal to some yn, where yn is of the form yn equal to k over root n. For two-sided level 2 alpha, with usual notation for the normal CDF, phi of k is then equal to 1 minus alpha. The problem is to find the distribution of m, which is the value of n at which the experiment stops. Now, if we denote by Fn-Sn the probability density function of Sn in the sequential procedure, then it is as is given on this slide. The density function Fn can be obtained recursively, F1 being the standardized normal density function. The probability of absorption at or before n observations, that is the probability that m is less than equal to n, is then given by Pn, and the expression for Pn is Pn equal to 1 minus integral minus yn to yn Fnu du. Alternative formulations can be based on the differences Pn minus 1. The basic method to evaluate these integrals is to evaluate them on a grid and to use numerical integration procedures such as Newton-Cotes and Simpson's 1/3 rule. The second paper by Pocock derives the parameters for group sequential designs essentially based on the results due to Agresti. And here we consider a clinical trial with two treatments A and B, a continuous outcome, and our statistic is di bar equal to summation j equal to 1 to I xaj minus xbj bar divided by I. This is known to be normal with mean delta equal to mu a minus mu b, where mu a and mu b are the means corresponding to the two treatments. The two-sided significance test of the null hypothesis, mu a equal to mu b, after I groups will have significance level pi equal to 2 into 1 minus capital phi root in di bar by root over 2 sigma. In table 1, we look at the nominal significance level and its corresponding standardized normal deviate for use in group sequential testing, where we have known variances for various choices of capital N and overall significance level alpha. So this table shows the value of alpha prime, which is the nominal significance level, and its corresponding standardized normal deviate for alpha equal to 0.05 or 0.01 and capital N in the range of 2 to 20. Note that for alpha equal to 0.05, n equal to 11, the nominal significance level alpha dashed is equal to 0.01. Table 2 shows the value of the standardized difference for normal group sequential tests with various values of alpha, beta and capital N. The main use of this table is to obtain suitable values of small n and capital N given information on alpha, delta and 1 minus beta. Note that for any chosen value of capital N, small n is equal to twice delta sigma by delta whole square, so it is convenient to express delta in terms of sigma. In table 3, we look at the average number of groups to termination of a trial under the hypothesis mu A minus mu B equal to root 2 delta sigma by root n, where delta is obtained from the previous table as a function of the maximum number of groups n, the overall significance level alpha and power 1 minus beta. Note that the average number of patients per treatment under the alternative hypothesis delta equal to 0.5 sigma falls from 84 for the fixed design to about 56 for a 20-group design. In Table 4, we look at the maximum and the average number of patients under HA for several values of capital N using a normal group sequential design with alpha equal to 0.05, delta equal to 0.5 sigma and 1 minus beta equal to 0.9. The sequential design is often inferior to the group sequential design in the cases where capital N is equal to 5 or 10 or 20, in the sense that both the maximum and average number of patients are greater under both the null and the alternative hypothesis. Finally, Pocock also derives sequential rules for other kinds of responses. In the first case, this is done for normal responses, but with an unknown variance. In this situation, we would expect to have some loss of power. For exponential responses, here we assume that we have two samples from the same exponential distribution, and we look at the ratio of the sample means, which has an F distribution. Binary responses, so here we have a binary response variable, and our plan is based on the difference in success probability between the two rules. This is a situation which we shall consider in more detail in the subsequent module. And finally, on the Wilcoxon test, when it is not possible to apply any parametric rule to the underlying data. In this second module on sequential clinical procedures, we concentrated on the phenomenon of interim analysis for group sequential trials. Much of the literature in statistics is on sequential plans for the case where we assume that it is possible to analyze the data after every patient or every pair of patients accumulate. In practice, this can be difficult, and hence a more viable assumption is to use the design of group sequential designs. In a group sequential design, we specify two parameters, which is the maximum number of times which we will look at the data, which is the maximum number of groups, and the size of each group, that is, how many patients will be recruited after each interim analysis and before the next interim analysis is performed. We have seen that the effect of interim analysis is to increase the overall level of significance, and thus we have defined two concepts, the overall level of significance and a much more stringent nominal level of significance, which we apply at each interim analysis. There are different procedures for specifying the nominal level of significance. This could be constant over time, or it could also vary as a function of time, being more stringent at the start and less stringent at the termination. We have briefly looked at the iterative numerical procedures for calculating the plan and the average sample number, and seen that they are generally based on similar results for the continuous case as has been derived by Armitage in his 1969 paper. Thank you.